Hello, my name is Willow Kion-Crosby, and I'm a postdoc in the BarQuest lab at the Helmholtz Institute for RNA-based Infection Research in Germany. And today I'd like to talk to you about my project on exploring how the choice of network architecture impacts biological inference when analyzing bacterial transcriptomic data using autoencoders. Now, bacterial expression is often a response to environmental stimuli as well as environmental stresses, and is also often associated with bacterial pathogenicity. So we'd really like to understand the details of these regulatory networks, as well as understand how the differences in bacterial expression make a bacteria pathogenic versus non-pathogenic. So to address these questions, we turn to the analysis of transcriptomic data. As the choice of analysis tool, we turn to deep learning, which has seen a variety of applications in recent years, including genomic as well as transcriptomic data analysis. Specifically, we use autoencoders, which are a type of self-supervised neural network consisting of an encoder and a decoder network. The encoder network takes in a data point, such as an image, and compresses it down to a low-dimensional representation, typically around 50 numbers for us, at what is known as the bottleneck layer. The decoder then takes that low-dimensional representation and attempts to reconstruct the original image. So both of these networks can be trained simultaneously in a self-supervised manner, with the objective of making it reconstructing as similar as possible to the original data point. So how do we apply this to transcriptomic data? Well, we actually train our autoencoders on bulk RNA-seq data for the bacteria Escherichia coli. And then we take the decoder of each of these networks and activate the neurons at the bottleneck layer and see which genes turn on and which genes turn off. These then define gene groups associated with each of these neurons, which gene set enrichment analysis can be performed on. Finally, we can take the original data set and pass it into the encoder of each network and see the patterns of neuronal activations, and such that we can associate the different experimental conditions with different biological pathways. So back to the original question, how does the choice of network architecture impact biological inference? To approach this question, we've actually looked at a variety of 7 different architectures which vary in network depth as well as overall structure. We've further looked at a variety of network architectures and varied the bottleneck layer, or the overall compression of the networks. So in general, what we see is that the deeper networks infer more biology than the shallower networks. In addition to this, we see that the deeper networks explain the data equally well in terms of generalization score, the test loss, with fewer terms. So fewer pathways are required to explain the same data points. But what about the network compression? Well, we actually see that as we relax this compression, the networks learn less biology out of the data. And this is in sharp contrast to the fact that as the bottleneck layer is expanded, the generalization score actually improves. So really this suggests that there's a balance, there's an optimal compression for each network, for each data set, which is a balance between total inference and generalization score. Finally, we see that within each of these neurons, or each of these bottleneck nodes, the gene groups which are defined there are easily functionally related, so they can be easily validated as biologically relevant to each other. We see that for nodes associated with iron uptake, as well as for flagellum assembly. So with all of this background, we then analyze a UPEC data set for Escherichia coli strains associated with a urinary tract infection. So within this data set, there are 5 isolates, strains from patients with known pre-existing UTIs. These 5 strains are cultured in lysogeny broth, and additionally in urine from healthy donors, so donors that don't have pre-existing UTIs. And then the original isolates are also directly sequenced, so that there can be a nice comparison between pathogenic and non-pathogenic conditions. So what do the autoencoders tell us about these data points? Well, first we can pass the original data set into the encoders of each network, and again look at the patterns of neuronal activation. And one thing that we see is that there's a group of neurons which solely activate during UTI versus the other two conditions. So within these neurons, there are genes which turn on during UTI and actually turn off during growth in urine. And within these gene groups, we see cold shock proteins as well as a variety of other genes which are associated with stress response, including antibiotic response. So in summary, what we've done here is explored how the choice of network architecture impacts inferred biology when using autoencoders to analyze bacterial expression data. We really see that the deeper networks infer far more biology than the shallow networks, and in more of a concise way, so with fewer pathways to explain the data points. We also see that there's a balance in terms of the network compression between generalization score and total inferred biology. Finally, we've leveraged our understanding of how the choice of network architecture impacts biological inference and analyzed a UPAC data set, and as a result of that, we see that there's a gene group that's associated solely with urinary tract infection. So thanks for your attention, and for any of the further details on this project, please visit poster 466 on July 13th.